animals into the food chain. So from an animal scientist perspective, we consider genetic engineering of animals as a basic definition of direct manipulation of an animal's genome. And that can be from traditional selective breeding, where we're bringing together the genome and the sperm and the egg, come together and create a new combination of genetics that may not have arisen in nature without human intervention. Or it can be sophisticated, like using cut and paste genetics with gene editing technologies. And so I thought I'd take a quick step back and just define what a genome is. And a genome is the list of instructions on how an organism is made, functions. It's a combination of nucleotides that are considered as letters. These make up DNA. They're packaged in chromosomes. And the complete set of genes encode for the molecules in this genome control traits. And so we and others have this grand objective of how do we engineer resiliency traits in livestock through genome editing. And I'm asked oftentimes by the general public why. And so I thought I'd provide some context from a scientific perspective. So the why for me is in addressing food security and insecurity. And so in developed countries, we often don't think about food insecurity, and that is because greater than 2 billion people on the planet are considered overweight. It's 35% of the world population, and over 90% of these people live inside high-income countries. There's also roughly 1 billion people on the planet that are considered malnourished, even though this is only 15% of the world population. Almost 100% of these people live outside high-income countries. And if you put the data together, more people on the planet die every year from starvation or malnourishment than cancer, malaria, foodborne illness, or HIV/AIDS combined. And yet in the United States, we spend 100 times more funding on research and trying to address these other areas than we do food insecurity. And so we have a global food challenge in addressing today's food insecurities, but also in going forward. What I've plotted here is the number of people on the planet from 1900 with a prediction out to 2050 based on historical growth rates. Somewhere around 1940, the world's population, human population, began to double every 40 years. So between 1940 and 1980, it doubled. Then between 1980 and 2020, it doubled again. And based on those rates, we're estimated to hit 10 billion people on the planet somewhere around 2050. The reason 10 billion is important is because at that point, we will need to increase agricultural output, both plant and animal, by 60% just to provide today's nutritional standards for 10 billion people. And simply putting more plants in the ground and putting more animals out to graze is not the solution. What I plotted here is the percent of unutilized land as either crop production or grazing land. So this is unutilized arable land. You can see in 1950 there was a good portion of unused land for either crop or animal production. But by the time we're estimating to hit 10 billion people on the planet, roughly 97% of all grazing lands worldwide will be in use, and almost 90% of all crop. So we can't simply put more plants in the ground and graze more animals. And so this is a global problem with increasing demand for agricultural products for food, but decreasing availability of agricultural land. And animal protein continues to demand worldwide. Between 1970 and the year 2000, it rose by 70%. Between 2000 and 2020, another 50%, and is predicted to continue to rise up to 2050. So animal protein isn't going away as part of the human diet. And so we operate on the belief that a solution to this global challenge is to enhance the efficiency of livestock production by building resilience in animals. And what I mean by efficiency through resiliency is how well animals can convert the inputs, such as water, both the amount and quality of water, the strategies that we put into them for disease prevention, like antibiotic use, and the feedstuffs that we provide to the animals, how well they can convert those inputs to measurable outputs in the form of meat, milk, and fiber. And of course, we're asking the animals to do that in changing environments, harsher environments, and to do it with having less impact on the environment themselves. And the one intrinsic variable of the animal that can influence how well they can convert the inputs to outputs is the genome. And so selective breeding is an ancient practice, in both plant and animal agriculture to impact genetic gain, the improvement in phenotypic traits from generation to generation by way of selective breeding. And so even though selective breeding can have a positive impact on genetic gain, it's not perfect. It's based primarily on observable phenotypes, which may not be combining genetic material to make good genomes. It also takes 5+ generations and 10+ years in animal agriculture to make incremental changes. We can also have genetic drift and inadvertent selection of disease states or bad phenotypes when we use selective breeding. And so the promise of genome engineering is that it offers a means to build resiliency, to enhance efficiency in a short time frame and without the inadvertent negatives. There's been conventional strategies to achieve this in animal agriculture, and they all rely on the use of recombinant DNA. A foreign piece of DNA that could not have arisen in nature. It is something that human intervention has to happen in order to bring together different combinations of DNA. This recombinant DNA has been attempted to create transgenic, genetically modified organisms in animals through a process called chromonuclear injection. This is simply after an egg is formed, there's a nuclear material from, when an embryo is formed, there's a nuclear material from the egg, and the nuclear material is delivered by the sperm. And if you can find those, and put a needle in it and inject the recombinant DNA, every once in a while it ends up in the genome of an animal. Well, it turns out this was really easy for simple organisms like mice, and it just doesn't work for large animals. The other is cloning. Put recombinant DNA into the nuclear material of a cell in a culture dish, and then take the DNA out of an egg and transfer that nuclear material. Cloning had great promise with limited payout because of the developmental abnormalities. And the last is the attempt to use embryonic stem cells in order to make genetic modifications, reintroduce those into an embryo, and now make an organism. All of these conventional strategies have great promises, but they really have limited payouts and never caught on. And so the promise of gene editing is that we can use molecular tools along with DNA sequence information to generate efficient and precise genetic engineering in animals, do this in 1 to 2 generations, and do it in a recombinant DNA-free system. There's been several strategies that have been developed for gene editing that can be applied in animals. The most popular is CRISPRs, and I'll tell you on the next slide why. The importance of all these strategies is that they can be done without the absence of foreign DNA sequence, or in the absence of foreign DNA sequence. And so you can bring about genomic changes that can endure a rise in nature. CRISPR is the most efficient and it's applicable to all mammalian systems. It's simply finding a spot in DNA that we want to make an edit, design a CRISPR strategy, which involves a guide RNA that tells where this Cas9 protein should go. What that does is it breaks DNA. When that DNA is put back together with the natural DNA repair process, it creates an edit. And that edit then can be passed on inheritably through reproduction. So if we want to make a genome edited animal, we simply put CRISPR reagents into a single cell embryo, create a pregnancy off of that embryo, and the entire organism is going to be a gene edited founder animal. And what we're looking for is that the germplasm in the sperm and eggs has the edit and can be passed on to the next generation by reproduction. And so CRISPR has really become the tool in the animal biologist, animal scientist toolbox to get genetically, what I would call genetically enhanced food animals, trying to replace engineered things. And so how I define the leading edge of application of genome editing in food animals, to define the future of food security, is in 5 bins. The first is improving the welfare of the animals. The second is developing disease resistance. The third is improving the growth and efficiency of growth and palatability of the product. Fourth is environmental adaptation, whether it's adaptation to hot climates or cold climates or drought conditions. And the last is reproduction. The efficiency of reproduction, because at the end of the day, genomes need to be passed from generation to generation through reproduction. And so an essential element in applying science to genome editing applications of animals is to be able to get efficient dissemination of the engineered genetics into livestock populations. If you think we make a single animal, how do we get a single animal and the genetics of that single animal into populations of thousands of animals? And that's where reproduction is essential. And so reproductive technologies increase access to unique genetics because germ cells link generations. And so if we have technologies that can disseminate on a large scale, either embryos or sperm, we can efficiently disseminate designer genetics or enhance genetics. And so we target the male side because each one of these sperm, and millions are produced every day, has a unique genome. And so we can achieve widespread access and large-scale dissemination of engineered genetics through the male germ line. And so one of the tools that's been developed and was commercialized in the 1960s is artificial insemination in order to gain access to elite genetics or desirable genetics. Simply, you can take sperm from a genetically desirable animal, and say it resides in North America, put it in a frozen state, ship it around the world for artificial deposit. So although it's an effective tool, it's not widely used in a lot of livestock production settings. In beef cattle production, it's a logistical challenge to apply artificial insemination, and so roughly only 7% of beef cattle globally are bred with artificial insemination. In swine, sperm cryopreservation is inefficient, and that limits the ability to get widespread dissemination of genetics. In small ruminants, it's a technical challenge. It's a surgical procedure to efficiently apply artificial insemination, and so less than 1% of small ruminants are bred by artificial insemination globally. And so one of the solutions that we've tried to work on is developing what we call surrogate males. They can produce the sperm with genetically engineered DNA. They can be disseminated by natural mating. So it's the technology we call surrogate sires. It's CRISPR engineering of reproductive capacity in livestock, and we've worked across the spectrum of both ruminants and monogastric livestock. So the concept I'm going to show you here briefly is I'm using cattle, which you can plug and play any species you want into this system in any scenario. If we have a donor animal that could be a CRISPR-edited founder with enhanced genetics, it could be a desirable breed, it could be an individual within breed that has elite genetics. The concept is the removal of testicular tissue. Sperm production is a stem cell-based process. So by isolation of sperm-producing stem cells, we can expand them in a culture dish, or we can freeze them, and then in concept transfer those into the testicles of surrogate recipients. And this could be a battery of tens or hundreds of recipients, and they can simply breed by natural mating and pass on the donor genetics. There's two key elements to this. One is the in vitro expansion of the donor stem cells, and the other is how do you make these recipients germline related so they can't make their own sperm. They're only making sperm of the donor. And so we've tackled this really difficult question of how do you generate male livestock that don't make their own sperm, and it was through CRISPR editing where we can knock out key genes that are needed for germ cell development. And by doing that, we can get a sterile surrogate that has functional testicles but can't make their own sperm, but can still be a host for sperm-producing stem cells from another male. And so we've targeted this gene called NANOS2 because we knew from lower order organisms like flies that have a NANOS gene that's knocked out in male and female sterility. In mammals, in mice, as the first model we worked on, they have 3 NANOS genes, the NANOS2 gene. In particular, only males are sterile, but females are still fertile, and we've shown that that's evolutionarily conserved in livestock now as well, in that when we knock out the NANOS2 gene by CRISPR editing, we have just male sterility and female fertility. So we've done this a number of times in my program at Washington State University, where we've been able to use CRISPRs to engineer males that are NANOS2 knockout. They can't make their own sperm, but we perform these sperm stem cell transplantation, and they start making the sperm of the donor. And so we're working globally to try to implement this as a breeding strategy in various livestock production systems across the planet. What you'll see is we aren't working in an EU member state. And the reason that we're not is because of the challenges we have with trying to apply genome editing into a food production system. And so I'm going to finish up by talking a little bit about my experiences with trying to advance a biological proof of concept in livestock production into public impact. And when we think about this as a winding road, we often don't see all of the curves and bends that need to be navigated in order to go from a basic discovery, technological advance to actual impact. One of the first things that we need to address is whether we're truly addressing a societal issue and a need with the science. And Considering that we are, the next phase is often a commercialization phase on how do we scale up to be able to provide it to producers. And then we have to address the public perception and acceptance and governmental regulation in order to get the impact. If we can't address these issues, everything stops here. So the science never gets out of the lab. And so over the last several years, I've had a series of federal and public engagement experiences in the U.S. system that have given me a perspective as a scientist and a developer of genome editing applications in livestock. In 2016 through 2020, and hopefully coming up now in 2023, we're going to have some more publications on gene editing applications in livestock through the scientific peer-reviewed literature. What this resulted in was a need for me to have an investigational new animal drug files with the FDA, which I don't need to do research, but I do need it if I ever want to advance the research out of the lab. And so we've been sharing information with the FDA for many years now. As part of the U.S. Academic Task Force on Gene Editing and Livestock Policy Development during the COVID years. In 2021, I testified before the U.S. Congress on genome editing in livestock. And in 2022, as part of a Foreign Ag Service workshop on gene editing in livestock in Brazil. And then in 2023, we became the first university to be granted food use authorization by the FDA. And so we were able to process some of our nanosphere gene-edited pigs for human consumption, and we made sausage out of it and fed it to people. And so in doing that, not only did it give me opportunity to engage with the media, but also in public forums about safety and the concerns. And then later this year, there's going to be a National Academy of Sciences in the United States that has been assembled to develop considerations for heritable genetic modifications in food animals. And so I'm going to leave you with some perspectives on the challenges of achieving impact of livestock gene editing. And the first one that I'll mention is the public acceptance piece. The GMO stigma is still an issue with the public. Even though the science and the intent of gene editing is different than conventional GMO, when the conversation comes up with the public, it's automatically dropped into the GMO pit. And so there's a need to have a new narrative, build a new narrative with the public, and I think we can do that through bioethical considerations and also developing better science communication skills. The second one I'll mention is regulatory policies, at least in the United States. It resides right now with the Food and Drug Administration, and it's antiquated. It's a regulatory framework that was developed in the 1980s for transgenics and recombinant DNA. Even though there's been a little bit of movement, it's still misaligned to the science. And so there's a modernization of the regulatory system in the U.S. that is hopefully going to be a combined effort between the U.S. Department of Agriculture and the Food and Drug Administration that makes it streamlined and cost-effective. And it's encouraging to see DEFRA making movements in the UK to modernize their framework. And hopefully the United States builds off of that. And so lastly, I just want to leave you with some conscientious considerations as a developer of genome editing applications on what the risks are and what a risk assessment might look like. The first one I'll mention is animal welfare. And the animal welfare issue is based on unanticipated negative phenotypes that might arise from a genome editing. And so careful design and beta testing in model organisms can eliminate most of the animal welfare issues. I think vetting of the phenotypes can be done at the research and development phase. So when we come into the commercial or societal phase, it's already been addressed. I think that animal welfare concerns with off-target editing or unanticipated phenotypes is really self-regulating because producers and developers aren't going to take animals that are unhealthy forward when we're developing it as an application. And then at the management level, both selective breeding and standard husbandry practices also impact welfare. And so whether genome editing is going to change that and reduce the welfare of the animal, I'm unsure. And then lastly, the food and the environmental safety concerns. That CRISPR-Cas9 DNA changes are going to alter the composition of meat and milk. And I think it's difficult to parse out the difference between naturally occurring mutations that happen during every replication of DNA, during every cell cycle, during every reproductive process. I think it's difficult, if not impossible, to parse out and separate the differences in composition that can happen through just normal mutation. The concern of off-target edits, just random mutation during cell divisions, also create changes to DNA. How to distinguish these and whether it's necessary in genome editing applications in animals is something that I think needs to be considered. And lastly, the environmental impact. What if the gene edit advances to the wild populations or feral animal populations? This is absolutely something that needs to be addressed and considered when doing a risk assessment. And so, lastly, I just want to give my opinion of what a GMO trigger in livestock production should be. I think livestock should not be considered GMO unless they have the incorporation of foreign DNA into the genome. They cannot arise without human intervention. And so bottom line is that animals with genome edits that are changes in DNA that could arise in nature should be generally regarded as safe. In my opinion, as a developer of genome editing applications, there should not be any labeling and traceability because I think it's almost impossible to implement and it's unnecessary.